Let us see the details of protein. Like nucleic acids, all proteins are polymers, but instead of nucleotides, amino acids build the proteins. Proteins have been called the foot soldiers of the living cells. Proteins' functions are the most diverse of all types of macromolecules. Protein varies in size, shape, and composition, which generate a wide range of functional properties. Some proteins carries out structural functions, whereas some speed up the chemical reactions. Each protein molecule has a unique three-dimensional structure determined principally by the amino acid sequence. Proteins are the polymers of amino acids joined together by peptide bonds. 20 different amino acids make up all the essential protein on the earth. What is amino acid? Amino acid has a basic design composed of a central carbon to a hydrogen. That carbon is called as alpha carbon. Then a carboxyl group, amino group, and a unique side chain or R group. The characteristics that distinguish one amino acid from another is its unique side chain, and it is the chain that dictates an amino acid's chemical properties. The unique side chain confer unique chemical properties on amino acids and also directive about how each amino acid interacts with the others in a protein. The amino acid can be classified as hydrophobic or hydrophilic and uncharged versus positively charged or negatively charged. Ultimately, the three-dimensional conformation of a protein and its activity is determined by complex interactions among side chain. Next, what is peptides? Amino acids are covalently bonded together in chains by peptide bonds. If the chain is short, that means less than 30 amino acid, it is called peptide, and the longer chains are called a polypeptide or proteins. Peptide bonds are formed between the carboxyl group of one amino acid and the amino group of the next amino acid. Peptide bond formation occurs in a condensation reactions involving loss of a molecule of water. The head-to-tail arrangement of amino acids in a protein means that there is a amino group on one end which we call as amino terminus or N-terminus and a carboxyl group on the other end which is called as carboxyl terminus or C-terminus. The carboxy terminal amino acids corresponds to the last one added to the chain during the translation of the messenger RNA. There are different levels of protein structure. To understand the basic principles of protein structure and the potential of its use in various areas of research, academic or industrial, we need to look into the way the 4 levels of protein structure that are related to each other. We'll see one by one the different levels of protein. Number 1, that is the primary structure of protein. Proteins are made up of a polypeptide chain in which amino acids join together with peptide bonds. The unique sequence of amino acids that make up a protein or polypeptide chain is called the primary structure. Peptide bonds are created by enzyme catalyzed condensation reaction and broken down by enzyme catalyzed hydrolysis reactions. Breaking down protein is important in many areas of the body, just not only in digestion. There are 20 different types of amino acid. So, for a simple dipeptide, there are 400 possible combinations, 8,000 combinations for a tripeptide. Obviously, the number of possible combinations is almost infinite when larger number of amino acids are combined to form a polypeptide. Second level of protein structure is a secondary structure. After synthesis, polypeptide chains are folded or pleated in different shapes called secondary structure. Secondary structure is held together by many hydrogen bonds, which overall give the shape greater stability. The polypeptide chain can fold back on itself in a number of ways. Each bond in the backbone formed by alternating amino acid central carbon and the peptide linkages which can rotate completely, and so a number of shapes are possible. It will become stabilized in a position where amino group become close enough to form hydrogen bonds with carboxyl group on another peptide bond in another chain or further along in the same chain. Often, these weak interactions between several successive amino acid residues result in a coiled structure, which we even called as helical structure. This is possible because of a result of studying X-ray photographs and constructing molecular models by Linus Pauling and Robert Corey in the year 1951, who proposed the protein structures were either in the form of an alpha helix or the beta pleated sheets. In the alpha helix, the polypeptide chain is coiled tightly in a fashion of a spring. The backbone of the peptide forms the inner part of the coil, while the side chain extends outward from the coil. The helix is stabilized by hydrogen bonds. 1 turn of the coil requires 3.6 amino acid units. The helix can be either right-handed or left-handed in the sense of threads on a screw. The naturally occurring alpha helix found in the proteins are all right-handed. Not all proteins have a helical structure, since some do not have at all, and they are random. The beta-pleated sheet is another regular secondary structure, as seen in the protein fibrin in the silk. In this structure, individual protein chains are aligned side by side with every other protein chain aligned in an opposite direction. The protein chains are held together by intermolecular hydrogen bonding, that is, hydrogen bonding between amide groups of two separate chains. This intermolecular hydrogen bond in the beta-pleated sheet is in contrast to the intramolecular hydrogen bonding in the alpha helix. The hydrogen on the amide of one protein chain is the hydrogen bonded to the amide oxygen of the neighboring protein chain. The pleated sheet effect arises from the fact that the amide structure is planar while the bends occur at the carbon containing the side chain. Next level of protein is the tertiary structure. Third type of structure found in protein is called tertiary protein structure. The tertiary structure is the final specific geometric shape that a protein adopts. This final shape is determined by a variety of bonding interactions between the side chains on the amino acids. These bonding interactions may be stronger than the hydrogen bonds between amide groups holding the helical structure. As a result, bonding interactions between side chains may cause a number of folds, bends, and loops in the protein chain. Different fragments of the same chain may become bonded together. The types of bonds which are included in the tertiary structures are ionic bonds or salt bridge that occurs between acidic negatively charged and basic positively charged side groups. Next, hydrogen bonds between the carboxyl group and amino group and the hydroxyl group on side chains. Hydrophobic forces between non-polar side groups. The other, disulfide bridges. Strong covalent bonds between thiol groups on pairs of the amino acid cysteine. Covalent links to other group such as the iron-containing heme unit in myoglobin and hemoglobin, or to the other types of molecules like carbohydrates or lipids, which forms glycoproteins and lipoproteins. The last level of the protein structure is a quaternary structure. The quaternary protein structure involves the clustering of several individual peptide or protein chains into a final specific shape. A variety of bonding interactions include hydrogen bonding, salt bridges, and disulfide bonds, which hold the various chains into a particular geometry. There are two major categories of protein with quaternary structure. They are fibrous protein and globular protein. What are fibrous protein? Actually, the final beta-plated sheet structure of silk is the result of interaction of many individual protein chains. Specifically, hydrogen bonding on amide groups on different chain is the basis of beta-plated sheet in silk proteins. Other fibrous protein such as keratins in wool and hair are composed of coiled alpha helical protein chain with other various coils, which is analog to those found in a rope. Other keratins are found in skin, fur, hair, wool, nails, horns, scales, beak, and many more. The second type of quaternary protein is a globular protein. On the other hand, globular proteins may have a combination of the above types of structures and are mostly clumped into a shape of a ball. Major examples include insulin, hemoglobin, and most enzymes. Dear students, with this we end to an introduction of the structures of nucleic acids and proteins. Thank you.